This animal can survive 18 minutes with no oxygen at all. Not reduced oxygen. 0. Total deprivation for 18 minutes, and then it gets up and carries on with no apparent injury. It's a naked mole rat. And today I want to explain exactly how it does that, because the answer is a piece of metabolic engineering, and because there's a detail in the honest version that almost nobody mentions. First, why it needs this ability at all. Naked mole rats are subterranean and intensely social. A single colony can approach 300 animals, living in a network of sealed tunnels underground. Think about the air in that environment. Hundreds of animals breathing in a confined space with poor ventilation. Oxygen gets consumed, carbon dioxide accumulates. In their burrows, carbon dioxide can reach around 10%, which for a human would be lethal. So their evolutionary problem was not an occasional emergency. It was a routine condition of daily life. Now let me explain why oxygen deprivation is normally so quickly fatal, because it comes straight back to mitochondria. Your mitochondria run the electron transport chain. Electrons stripped from food get passed down a series of complexes, and at the very end, oxygen accepts them. That's the terminal step. Oxygen is the final electron acceptor. Take oxygen away and there's nowhere for the electrons to go. The chain backs up. Every complex fills. The proton pumping stops, the gradient collapses, and ATP production from oxidative phosphorylation ends. That happens fast, and it happens in every tissue at once. Your fallback is glycolysis, breaking down glucose in the main body of the cell without oxygen. It's the ancient pathway, and it works without mitochondria at all. The problem is that it doesn't work for long. Glycolysis produces lactate when it runs anaerobically, and lactate accumulation inhibits an early enzyme in the pathway called phosphofructokinase. That enzyme is a control point. Block it and glycolysis grinds to a halt. So the fallback shuts itself down. Energy production ceases, and in the brain especially, cells begin dying within minutes. In the same experiments where naked mole rats lasted 18 minutes, mouse heart rate declined continuously and was undetectable on the electrocardiogram at around 6 minutes. Alright, so what does the mole rat do differently? It changes fuel. When oxygen disappears, the animal releases large amounts of fructose into its bloodstream. Not glucose. Fructose, the sugar more commonly associated with fruit. That fructose is actively transported into the brain and heart and metabolized there. The researchers identified the molecular signatures, widespread expression of a fructose transporter called GLUT5, and high levels of an enzyme called ketohexokinase that handles fructose. And here's why it solves the problem. Fructose enters glycolysis at a different point than glucose does. Specifically, it enters below the step controlled by phosphofructokinase. So when lactate accumulates and shuts down that control enzyme, glucose metabolism stops. But fructose metabolism doesn't, because fructose was never passing through that step in the first place. The roadblock is still there. Fructose simply joins the road after it. That's the whole trick. Not a new pathway. The same ancient glycolysis, entered through a different door that isn't locked. While this is happening, the rest of the animal powers down. Heart rate drops within about 2 minutes from a baseline near 200 beats per minute to a steady 50, and holds there for the duration. They lose consciousness. Breathing stops. Researchers describe them occasionally attempting a breath while dormant, as though periodically checking whether conditions had improved. And when oxygen is restored, they resume breathing within moments and go back to normal activity. Now, the part that I think matters most for you. The genetic pathways required for heart and brain tissue to burn fructose this way are present in all mammalian cells, including yours. You have the transporter genes. You have the enzyme genes. Every component of this system exists in your body right now. Your cells simply don't deploy it that way. Humans do metabolize fructose, primarily in the liver, but we don't perform this emergency switch in the brain and heart under oxygen deprivation. So this isn't a case of a foreign adaptation we could never have. It's an existing set of parts, wired differently. Now the honest caveats, and there are two, and both are the sort of thing that gets stripped out when a finding travels. The first is scientific disagreement. A comparative physiologist at the University of Oslo, quoted in Science's coverage, cautioned that how important the fructose switch actually is for the animal's survival isn't clear, and pointed toward other mechanisms potentially contributing. That's a serious objection from someone who studies oxygen deprivation in animals professionally. The fructose switch is real and well documented. Whether it's the main reason for 18 minutes, or one contributor among several, is not settled. The second caveat is about temperature, and I think it's the more important one. Naked mole rats are poikilothermic, meaning their body temperature tracks their environment rather than being tightly regulated. Their preferred body temperature is around 30 degrees Celsius. That's roughly 7 degrees below yours. Temperature matters enormously for metabolic rate. Colder tissue consumes energy more slowly, which extends how long it can survive without resupply. The researchers tested this directly. When they warmed the animals to 37 degrees, human body temperature, maximum survival dropped from 18 minutes to 6. 6 minutes is still extraordinary. It's around the point where a mouse's heart had already stopped, but it is a third of the number in every headline. So the honest version is this. A naked mole rat at its own body temperature survives 18 minutes of anoxia. At human body temperature it survives 6. The fructose switch is genuinely real, and there is professional disagreement about how much of the total effect it accounts for. That's a less clean story than the one that circulates. I'd rather give you the real one. There's a connection to something I covered a few days ago too. I talked about red blood cells which have no mitochondria at all and run purely on glycolysis. Here's an animal that temporarily converts its entire body to that arrangement. Switch off the mitochondria, run on sugar alone, and wait. Two very different biological solutions, both arriving at the same place. When you can't use oxygen, you fall back to the pathway that never needed it. So what's the takeaway? There's no lever here. Nothing to do, and I want to be firm about that because this is precisely the sort of finding that gets turned into a supplement pitch or a breathing protocol within about a month of going viral. You cannot induce this switch. Nobody has shown that in a human. The pathway exists in your cells and it is not under your control, and anyone selling you access to it is well ahead of the evidence. Now, where my own recommendation sits, and today it's entirely separate. CoQ10 is a carrier in the electron transport chain, which is the system that stops working when oxygen runs out. Levels tend to decline as we age. That's a claim about ordinary aerobic energy production. It has nothing to do with anoxia tolerance, fructose metabolism, or anything else in this video. The link in the description below goes to a free 5-item morning checklist. Mitochondrial is on that page as well, and it's an affiliate link. I earn a commission at no extra cost to you. And if you want the version of the finding with the caveats left in, subscribe. This video is educational only, and it's not medical advice.